delivery. So those lipid nanoparticles, they can be used for different applications, like you can deliver chemotherapeutic drugs for cancer applications, you can deliver proteins, peptides also. So it can be used for targeting also diseases like targeting cancer. So
the lipid nanoparticles, as we know them, have established for liver diseases and for mRNA vaccines, and now we're looking for more larger RNA molecules and gene editing and several other applications. But as we grow our understanding about the biological barriers and the physiology of the disease we are trying to treat, I think these lipid nanoparticles will also need to evolve.
You can stick almost anything in lipid nanoparticles, and if you're choosing the mRNA, which can encode almost every protein, so you can go for vaccination, cancer therapy, it's just the possibilities are endless.
I think lipid nanoparticles offer a good solution for all kinds of therapies and indications, not only COVID vaccines, because it offers a whole open space for gene editing, for example. So not only treating the symptoms, but curing the cause of the disease.
So with the liquid nanoparticles, what happens is you can really hone in and focus in and really increase the kind of selectiveness of the oncology targets. So that helps because you can be much more higher affinity drugs, and the drugs can then be more effective than a typical traditional approach that we have now.
I see basically the main fields right now is, or main fields of interest in gene editing, for example, to use lipid nanoparticles to bring mRNA and guide RNA cargo to the regions of interest to help cure diseases there.
There's no limitation on the gene therapeutics. Technically, if you find the right way to deliver your gene and find the right way for it to working, there's very much massive potential to treat all sorts of diseases or doing prevention of diseases. So,
yeah, I think the biggest challenge for translating the work we are doing in the lab to manufacturing the lipid nanoparticles is the in vitro in vivo correlation. So.
With the LNPs and RNAs, I think it's truly establishing itself as a plug-and-play platform in a lot of cases, and this is really going to accelerate access to patients where they really need it. It's going to open up the field when it comes to precision and genomic targeting as well.
Stability and scale-up, I think that's the most challenge that will the industry maybe face in this area.
It's very important to consider the process as whole, because if you change any single parameter, it has an impact on the quality of the end product.
One of the key challenges in translating LNPs from laboratories into production spaces is to have a consistent methodology for producing them. So going from that really tiny scale through to large-scale continuous manufacturing.
And for the LNPs, for example, it's essential that you know that you have the right particle size at all steps of the process.
You have to think about the quality, because when we're using it in the batch and the lab, we need to think about what quality assurance we have for this batch when we move into manufacturing and scale-up applications.
There still exist a lot of challenges, though, when it comes to understanding the process parameters that control the particle product attribute and the quality of the product at the end. We still have a number of unit operations, especially upstream and downstream, which need to come in integrate much better with the core manufacturing.
So the processes for manufacturing of LNPs is obviously vital, as with any other process, any drug development steps. I think getting the actual kind of the validation and actual the robustness and comparability between batches is vital.
So I think a big pharma has a role to play when it comes to establishing a bit more on the commercialization, on the operational side of things.
Patch is a very critical attribute in drug development, manufacture, enables consistency, enables comparability. So having patch as a part of your process development is vital, and it's something that is quality driven and it helps when you come to submit, and regulators also value this approach you've taken.
If you employ process analytical testing or process analytical technologies in manufacture, what it really allows you to do is fail early. So if you're producing a medicine, you've got problems, the quicker you fail, the better the savings are in the long term.
During the COVID, most vaccines use LNP, so that's why LNP getting much more hot topic now. But instead of LNP, something like polymer-based vector or other materials can also do the similar job.
I think at the moment we're just starting to scratch the surface on how materials such as lipid nanoparticles can be used to deliver genetic materials and therapeutics. I think in the coming years we're going to see a lot of investigation into how these types of materials can be targeted to specific tissues or organs within the body, and how we might be able to piggyback on the body's own physiological mechanisms to direct those particles to other locations than the liver, where they typically accumulate.
I think in the next couple of years we will see many developments of lipid nanoparticles, but maybe also other particle systems going beyond liver.
What I think will emerge in the field of nanoparticles is definitely gene therapy, so to offer a solution for gene therapy related diseases.
Although LNPs offer a great insight into what we can do in terms of therapeutics and drug delivery, there's plenty of scope out there to have a look into these other nanoparticles as well.
So in the field of lipid nanoparticles, I believe a big focus and trend is to trying to identify and screen and synthesize new chemicals that's, for example, new ionizable or cationic lipid and other components as well, to enable particles can travel to where they are most effective, and to be more effective essentially.
So we need to teach new tricks to all drugs, because